Yeah.
Okay. We can check one more off the list.
Yeah. That's on page 2.
Oh, look at this. Look at this.
Oh, got a fishy. Oh, octopusy.
Oh, octopus.
Oh,
Hello.
Oh.
We're waiting for you, Dumbo.
Wow.
Dumbo.
Hello.
Dumbo.
Awesome.
Hello.
Hi, friend.
That is
Hi,
cute.
friend. I need
Oh,
to-
that's fantastic.
Don't come any closer to me.
Yeah, stay there.
Uh-oh.
Look.
Uh-oh.
Oh,
Oh, there's my
that's
eyeball.
beautiful. Look at how close.
That doesn't remind you of chicken skin, Wayne?
No. No. It does
Uh-oh.
not.
Uh-oh.
Beep, beep,
Oh,
beep,
No
beep,
grabbing
beep.
on.
Fly away.
It wants it.
I
That's
don't want to be
gorgeous.
that close to you. I respect your distance.
Oh,
Blown around.
little turbulence.
A little wrinkly.
Little turbulence.
All right. Enjoy your life without us.
Just wants to be
Well,
friends.
Hello.
Look at those cool modified
Okay then.
wings.
That was a
wonderful.
photo bomb.
That just totally,
Well, hello.
Tiny and Argus.
totally buzzed us.
He was real close. That was cool.
Oh wow. Alrighty then.
That's a great look at the texture, though.
I'm
right?
sure.
I just have to tear myself
Oh,
away
there's
from
the little candelabra ones.
I
octopuses.
felt like
octopus.
he was doing the exploring and
Exactly. We're the subject.
we were the
Yeah.
subject.
Learned a lot. All right, can we zoom in on this little
Sure.
candelabra-looking one?
What is?
I feel like this was in our guide.
Yeah.
Yeah.
Steve spoke about it earlier.
Yeah.
Is
Mm-hmm.
that on our?
Another bamboo coral.
Isabella, maybe?
Yeah, maybe.
Yeah.
That's what it
Can we
looks
go
like.
a bit tighter?
Go ahead, Wayne.
More?
Yeah.
All right. Oh, lost the focus. There we go.
That's great.
Wow. Another one with nothing
Maybe
on it.
Drop the light on it.
Yeah.
Looks like a menorah.
Yeah.
Mm-hmm.
Okay, coming wide.
All right.
Yeah, looks like an Isabella.
More light.
Bridge nav, another step.
Another photo bomber.
Shrimp this time.
Yeah, he's still there.
Left of Argus.
Oh yeah, you can still see the
Chris.
Dumbo in the Argus feed. Oh, that's
Dumbo.
super cool. Check out channel 2 if you're watching on the website. Just slowly making the way along the mountain. Out for a stroll.
How can you tell by looking if an octopus is a male or female?
Is this a joke or a
I
real
was just gonna
question?
say, that's not
Yeah.
the setup for a joke. That's a serious question. It did sound like the setup, didn't it?
I don't know, Trevor.
How do you know? Well, Wayne.
The males do have a little mating tentacle. I don't remember if it's a extra thing you can see, but when you see them mate, the male just kind of gets next to the female and slips a little tentacle underneath.
It's one of the corkscrew.
Oh yeah, these are cool.
Corkscrew, they were calling the toilet brush before.
Oh no.
Next, a bunch of those hydroids or those, or the little stolen ephorus.
Be cool with a step away from the wall next time.
Chrysa George there, I
Yeah.
think.
Iridogorgia.
Yeah.
Yeah.
Irida?
Iridogorgia. Uh-huh.
Let's have a zoom on that, please, Wayne.
Let's see
That
what
was
we
so
can do.
cool.
We had the
It's beautiful.
beautiful red ones earlier too.
Bridge
So
now.
cool.
Those do look like fireworks.
Yeah.
Mm-hmm.
20 meters, bearing 040.
Hi to Rachel Rayner, one of our science communication fellow
Hi, Rachel.
alumni and legends.
Yeah, there's a shrimp in there too, I think.
Oh yeah, there is. Hi,
Very
shrimpy,
cool
shrimpy, shrimpy.
associate. You can also
Cool.
see some of this scale. The Argus shot is so cool right now with this coral backlit
Yeah.
against that cliff. Oh, I love that.
Full wide,
Thank you.
more light.
It's a huge stock. I
Is
think
it-
some of these Iridogorgias are some of the ones
That
that have been aged to
I'm gonna come up the wall a
hundreds
little bit
of
just while
years.
Argus is cozy. Ooh.
Got a fish up on the ledge too.
Ledge fish.
I see you, ledgefish.
You can't hide.
Hey. Ebony,
We're ready
Ebony
now.
and ivory.
Piano?
No, it looks like the current's going down the slope here.
So interesting they would change.
I was moving up. Let me hold
Oh.
steady and see what way it's going.
Yeah, no, I'm just looking at the acrinoid, the orientation.
Oh, I see.
Oh, yikes. What's that green thing there?
Should I push in?
I don't see the green thing.
I
It's
think
right in the middle there. Or is that yellow, whatever it is?
I
Right
see a red thing.
under the lasers now.
Oh, yeah, under
Yeah.
the lasers, yeah.
Mushroom? Oh no.
Baby crinoid?
Yeah, it's a little acrinoid.
An unstocked crinoid. Cool.
And less light.
Hey, you're doing good, Wayne.
Okay.
Okay, come wide.
Coming wide. Whoa. Not with the focus.
It's hard to control zoom with focus.
No.
Nearly impossible, some would say.
Yeah.
Bridge nav, another step.
There's so many little cubbies and fantastic little spots here.
Cubbies, I like that. I feel like I'm gonna
Just
try
a
to store my jacket.
Chrysogorgia cubby. Yeah. Put your lunchbox in there. Just the extent of this feature is so cool. We launched this dive at midnight last night, got to the seafloor about 5 a.m. after sorting out
Let's zoom on this little guy, Wayne.
some
Okay.
technology.
Oh, we're gonna need a lot less iris, but I'll fix that after we get in.
Sure.
And we have been pretty much along
This
this cliffside the whole time.
I'm
I'm
kind
gonna,
of.
I can see if I can put it with the rock behind it to get some contrast there.
Yeah.
Wow.
Is that a hydroid?
Yikes.
Looks like.
Pretty cozy with the rock there.
I don't.
Yeah,
Or
it
another
is.
precious coral maybe? Could that be?
Yeah, that's. Whoops.
They're all precious to me.
This looks like the,
Alright, I
it
actually
looks
gotta
like
go.
the
Sorry, Wayne. I'm
Pleurogorgia
all
with
tucked in there.
Okay.
the polyps only on one side of the branches. But I feel like sooner we should put just an asterisk
green.
next to all
Coming
these
wide.
that I'm
Oh yeah.
I'm
for sure.
identifying them.
I feel like there's some more red action in there now. I don't know what the red's. Yeah, that's looking better.
Let's see. You want some red? There's
There's
a
another.
red coral.
Yeah.
On the
Oh, yeah.
stalk.
Upper right.
Yeah,
That's-
crinoid.
Crinoid?
Mm-hmm.
You ask for red, the sea delivers.
We can image that one as we go by.
All right, let's go in.
I've seen a lot of yellow ones, but not many of these red ones.
No.
Yeah, they were everywhere on our last watch, so back at the start of the ridge. This one looks like it has some hydroids along its stalk or something coming up.
Wild colors on that, eh?
Gorgeous.
It's a nice shot.
Yeah.
And
Come
about to
on.
hit.
Brittle star in the back.
Just missed.
Alright. Sorry. Okay,
Breach nav, another step.
a little more light.
So Rob, what were your takeaways from the parasitic cone, seeing that go by? Did you note anything different earlier in the dive when that was happening?
Not really too much. Just that there's a lot of pillow basalts on top, which isn't really surprising. So you went from the general carbonate reef sort of material on the very top to the pillow basalts. Then we crossed back over pillow salts into the manganese nodules in the flatter area. And I was kind of surprised that look, a lot of small pillows in the cross section, but then you got into some very massive flows too. So some very thick, steep cliffs, which I guess not too surprising, but it was interesting.
Did it look bisected like you had hoped?
No, I always expect to have like a cross section you see in a textbook with a
Yeah. Very neat little like layer cake outlines. Here you
Exactly.
see the 1921 eruption. No. Or the 21 million year ago eruption. So science is sometimes slow, but maybe in a year or two, we will have the ages on the rocks that we collected from both the base along the slope of this tumble down area, the mass wasting landslide type area, as we climbed the main feature of this seamount, and then also the rocks from this parasitic volcanism cone on the top, which you'd expect to be much younger if this was a feature that developed on top of the carbonate crust, and after a time when that was well established on the top.
Wayne, could you push in past the vignetting, please? Thanks.
So Rob, what's the sampling processing like for these rocks? Are we gonna break them in half with a hammer? What are we gonna do?
No, I think they're going to use the rock saw.
Okay.
Cut them open and see what's inside. Maybe take some slabs and for later processing and then archive the rest.
What's the blade made of for the rock saw?
It's actually industrial diamonds on it.
Okay.
It's pretty interesting. And you can actually put your hand on it while it's cutting and it won't cut you.
Wait, what?
Have you tried that?
Yeah. And,
'Cause it's an abrasive cutter, right? It's not a...
yeah, exactly.
That's wild.
I'm building 8 houses right now, so I'm like,
Right now?
I know a lot about power tool safety, and I'm like, no, that does not. It's not safe, everyone. That's not what
Do
you
not
do
try
with a
this at home.
giant chop saw, because it just looks like a chop saw. It
It's
looks like a
basically a
pivoted chop saw.
fast-moving emery board.
Yeah,
Yeah.
that's basically what
Right.
it is.
Just sands through it.
Interesting little lava pile here.
Yeah, this is cool.
See the predominant direction of the current here, with the sediment's collecting on the back side, on the lee side of this rock.
Hmm.
So it's kind of going from left to right on our screen.
Looks like it's kind of pouring over the edge.
Yeah.
Which is true, as we just taken over the watch. That's the direction the ripples were indicating, up over the top towards the northeast as we
I
traveled
might
out
pull you a
towards the
little bit.
edge.
Sorry about that.
Don't jump.
You're
This
too
is
much
so
to dive
cool.
for.
This is
No. Still young.
so cool. This is so cool.
No, don't do a full loop.
I'm doing it. Taking a wrap out, and might as well make it cinematic.
Yeah, no, just stop, pivot, turn. Sweeping. Okay, Rob, I love this question from a viewer. They're like, they've got basalts and they've got carbonate from your explanation. Ancient eruption, accretion, nodules. What's the time scale on nodules? How do nodules form? Why are they round? Can you talk nodules to us for a little bit?
I'll try.
Okay.
I mean, they're very slowly accreting, and they accrete. You know, they actually basically collect the minerals out of the water,
Mm-hmm.
and they precipitate, and they just kind of grow in circular, and they grow about 1 to 5 millimeters per million years. They grow very slowly, and so they're just round because they keep adding to the outside of it, and they typically occur in low sediment environments, which is surprising to see some of the ones on top of here.
Mm-hmm.
And it's surprising the
Bridge
composition
nav, another step.
does vary quite a bit
Okay.
from place to place. And like, I think the ones in this region are nickel rich, whereas other regions have more cobalt rich or other sort of special minerals that due to a region. I'm not really sure why, if that's just the local character of the water column or a different sort of process that happens at the sediment water interface.
So interesting. And these are a target of interest for deep sea mining, for rare earth mineral acquisition.
I'm seeing red.
Definitely. Yeah, definitely an emerging industry. A lot of questions.
Yeah, it's a nice little coral in there too.
Oh,
Yeah. Little pink one.
You want to zoom on it? Is that what we're talking about?
Yep.
Okay, I'll make that happen. Go ahead, Wayne.
Ooh.
Doesn't look like the bubble gum.
Where you're going to be.
No, it looks different. All right, that's as close as I can physically get. Can you zoom in a bit more, Wayne?
It's got an associate too.
Maybe a...
All right,
I
come
think
on.
like maybe a paragorgia, but I'm not sure.
Thank you.
Doesn't really look like Swiftia. Not sure.
There's some big pillows here.
Mhm.
Yeah, really big ones.
I'm gonna back off a bit and see what they look like. Get some more general geology overview here.
Yeah, it looks like right at the bottom of the screen you see the larger lobate flows and then smaller flows in between smaller pillows. Different intervals of eruption. then on top of that, you have a more massive layer. Probably more voluminous flow.
Is there any way to ever know how many eruptions there have been?
Yeah, I mean, some people on volcanoes actually can go down and date some of the layers. I mean,
Okay.
it's easier on subaerial volcanoes because they actually date the, oftentimes the soils in between. Easier
Oh,
to
yeah.
date. But you can probably take some of these rocks if they do have the right mineralogy and do the kind of work that Kevin does with potassium argon.
They were around 7, Trevor.
Around 7. Hilarious.
There's another one there, another coral there.
Yeah, I might be able to get closer to this one, actually. Well, we will see.
Bridge nav.
Okay, go ahead and zoom.
20 meters, bearing 000.
Yeah, question I don't know. Can we make manganese in the lab? I would expect not. I think sort of the...
Looks like another
The-
paragorgia.
yeah, it does. Maybe the advantage...
Hemicorallium, something.
Remember? Me. I'm
Thanks.
thinking paragorgia. I'm not sure.
I got a coral question.
Yeah.
You know how corals grow really old and really slow?
Yes.
If you had or if a coral had the perfect living conditions, like a good amount of flow, but not too much to knock it over, lots of food source, all this kind of stuff, how fast could a coral grow?
It's a good question. I think it depends on, at least, like, speaking about shallow water corals, it's really variable by species.
Oh, right, of course.
So I would just guess that deep sea ones are the same. Some can grow, you know, you can see them grow over a season, so you could grow a couple centimeters a year on really fast growing corals. There's a lot of hope for microfragmentation of shallow water corals right now, that they're learning that actually by breaking corals apart, there are some sort of mega boosts. They sort of put, you know, growth hormones and processes and the protein building, just sort of protein synthesis and tissue development into overdrive, and they can get corals growing really quickly. I don't know, but I want to ask Steve that question now. Wake up, Steve. We have questions for you.
We see them growing several centimeters per year in the photic zone.
Yeah.
You know, they have something a lot of well, American small they call it Tai Sama Sama, where it's basically translate to stinky tide, and it happens every year when we have these high highs and low lows, that the corals just kind of grow throughout the year, and when you get these super low tides, they die on the top, and it's like mowing the coral down. That happens every year, so that's why you kind of see them right at that tidal interface, growing right up to that tide mark.
Yeah. Yeah, that's a good question. Some more nodule questions coming in. Rob is distributing snacks throughout the van right now, but deep sea mining is a infant industry around the world right now. It is the reason people are interested in deep sea mining. So the questions about, can we get these chemicals from the lab? Can we make them synthetically? Currently, the sources of rare earth minerals are concentrated in a few land-based deposits around the world. And the reason we need these are rare earth minerals are really important in technology, in manufacturing circuit boards and microchips, and all of the technology devices that you're listening to Nautilus Live on right now and that we carry around. Rare earth minerals are also really important in battery technology. There are a massive amount of metals in solar panels.
This is
and
a
in
long,
wind turbines.
sorry to interrupt you, this is
No,
a
go
really
ahead.
long coral coming out. Look at it on Argus, that is really long.
Oh yeah, I can see.
Wow.
I don't want to slide into that. I almost would have taken that out.
Holy
So
cow.
Ooh.
that is a full Herc-sized limbo bar.
Oh, that's cool. Yeah,
Like, we're not
let's
even
make
close
sure
to
we
it
get
yet.
get all of it in the image.
Yeah, can I have bubble cam on number 4 or 6 or something, whatever the Mongo is?
Wrong arm.
Other one.
Trevor senses a challenge.
Yeah, perfect. Can I have it seeing more of the front there? Just so I don't run the lens into the rock is all. Great, thank you.
You might, there's another one, a few in the background.
What would you guess how long this could be?
Wayne, could you
Bridge
zoom in?
nav,
I
another
can.
step.
Well, it's extending all the way back behind Hercules right now in the Argus view, so that's got to make it
You
at
want
least
to kill the
3
lasers?
meters.
Sure, we can kill the lasers. All right, I'm gonna back off now.
All right. And I'm gonna
Follow it down. You stay in. Yeah.
Keep it in focus and keep the lighting consistent.
Oh, a little fast. Defensive mode. You can see as it's channeling or kind of moving along like a wave along the thing. That was really cool.
Unfortunately, those are what I use to focus, the little fingers. I'm like, now I can't tell.
Bring your bulbs back out.
Can you see any knots? Can we say this is a bamboo coral?
There might be one.
Oh yeah.
There they come.
Yep.
That's really cool how they open and close as a wave.
Yeah.
A little message passed along the coral.
This is at least 3 meters.
At least.
But wait, there's more.
At least 5.
Well, we could see how far the front is from the wall, right?
Yeah.
Yeah. Can you come full wide? I'm going to put it. What I'll do, I'll tell you how big it is. I'm going to touch it with my sea prints. And then I'll tell you how far away the wall is.
Perfect.
Can you brighten it up a bit, please, Wayne? It might look a little ugly, but yeah, it's good there.
Oh yeah, you can measure it with your sonar.
Exactly, yeah. The old sonar measurement tool. Oops.
Bingo.
Ooh. How far away am I?
First division.
First division, okay. Beep, 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 beep. That would be 4 meters.
Wow.
Wow.
So
That's
cool.
awesome.
That's a big boy. Right on. Maybe we'll keep this here. That's actually probably more useful than front porch right now. And unless you want to do gauges, then steal it away.
Okay.
Hello to Dean Pettenchef, who from Los Angeles Natural History Museum, tuning in on the Scientist Chat and joining us online. Thanks for staying up late with us, Dean.
Trevor, at some point I want to just zoom in on just an area of the rock that we don't see anything, just to see what we can see on kind of a microscopic level. Kind of like what they did with Hubble. You know, they pointed Hubble at a black place of sky and they just
enhanced. Yeah.
and they found lots of stuff.
I'm a big fan of that stuff. I love that in mud transects where it's usually really boring. I like taking the time out, taking 30 seconds or a minute to get a good mud zoom, which you'd think is otherwise
you're not going to see anything.
And it's amazing how much life you find and all sorts of cool treasures and goodies. So yeah, I'm definitely into that. And maybe we see nothing, but then we know that.
Mm-hmm.
Yeah.
That's what exploration's all about.
If we knew what we were
And
going
come
to
up the
find, we
wall a
wouldn't
little
need to
bit.
come here.
Yep.
How about this blank face? How about this one?
If we're going to be here a while, I can zoom in on it.
I don't know that we're going to be here a while. I don't think we're going to be anywhere for a while, but we can be here long enough to
to check it out.
Stay wide for a sec.
Oh, sorry.
Careful your head.
Thanks for the treats, Rob.
Yes, thank you.
Did you notice the name on it?
Zoom in, Wayne.
Okay, zooming in.
Masterpieces?
No. What type was it of chocolate?
Belgian?
Belgium.
No.
Godiva.
Godiva?
Godiva chocolate.
No, you did not.
No.
Yes, I did.
Did you just stand in the candy aisle and survey all the options until that came to you?
I'd never planned things like that.
You know, Wayne really likes puns.
I heard he does, yeah.
Stop it. Yeah.
There's that gray sort of encrusting sponge that just in the very upper right corner that we.
You get any more zoom there, Wayne?
That's it. That's all I got.
All right, that is full me physically zooming too because I am against the wall.
Yeah, it's pretty steady too.
Yeah, that's cool.
But there's
Is
a
it-
lot of little things to
Yeah.
inspect.
Are those little white wormy things, are they animal? Or are they just detritus?
They might just be detritus that got hooked.
Well, I mean, Steve was actually saying there's some benthic forams that do that sort of thing.
Cool.
Yeah,
Yeah, and there's a few that have little branches and.
Okay, come wide, please.
Okay,
I'll move
come
along.
wide.
Super cool.
Good call on making time for that, Wayne. I think that's
Yeah.
valuable.
Okay. And punching in. Well, hello.
That's nice.
They're fun when there's these fun things everywhere. This is a good seamount.
This is a really good seamount. I love this seamount. It doesn't even have a name yet. The seamount west of Kingman.
I dove the ocean on a seamount with no name. Any young fans? All right.
Is that a mushroom or an anemone?
Oh, mushroom.
Mushroom. Fungus.
Can you still bubble cam?
Yeah, go ahead. There's a question. You don't really see plant life past, correct me if I'm wrong, like 100 meters or so. After that, it's all animals. Do you see fungus in the ocean at any significant depths, or is it all surface stuff that has gone down?
Corals can get fungal infections, I believe.
Really? Fungal infections on coral? Okay.
Shallow water ones, yeah.
But not the deep water ones?
I haven't heard of a deep water one getting one.
Because fungus, again, I'm not really much of a biologist, but fungus is not photosynthetic at all. So does it need light to live for some other reason?
No, it's a consumer.
So couldn't it consume at depth too?
Presumably.
So I wonder why we don't see it.
Science, good question. Science.
Bridge nav, another step.
Yeah, I don't know that there are really a lot of obligate marine fungi.
Good idea, yeah.
I mean, they do have records of fungus in sediment cores.
Is that an urchin at the near the bottom center? Oh no,
Anemone, I think.
anemone.
That's interesting, down in the sediment.
Yeah, and for a while, when they did the analysis, we think they over-reported because a lot of what they were reporting was a fungus that's similar to what grows on dandruff.
On what?
Dandruff.
Sure.
So it's human contamination that they were analyzing.
Okay. Yeah, I'm reading that it's interesting. Fungus that grow in the marine environment aren't a taxonomic group. They're mostly just a shared habitat. So there are a variety of ones that, you know, maybe adapted or be living in this environment, but it's not like a lineage of fungus. Fungi. Funguses.
She's on a different watch.
All right, come on.
Really thought
Yeah,
we'd see banks of
I
coral
want to see what
here.
the humidities have been for the bottles.
That'd be
how
nice.
they've changed over throughout the dive. Have they been pretty constant? Okay. Temperature, bottle temp's been constant? Is that the whole dive or just this sheet? Okay. Is it... the first reading is the one I'm most interested in because the temperature is probably still high then. Okay, and okay. And then of course the temp dropped, humidity went up. Great. And how about the Argus one? Similar. The starboard one's the one I'm most interested in.
Bridge, no?
Center screen, there's
Okay.
a tiny little thing.
You're 20 meters,
see that down
bearing
10.5 and
050.
Okay, weird. Okay, thanks.
Trevor, you've totally nailed a cutting edge topic. May of 2018, Woods Hole hosted and MBL hosted a marine fungi workshop to address a largely unstudied portion of the marine environment.
Fungus.
And there's a whole, there's a
Wow.
whole report about it. Some of the challenges are identifying fungus that are truly marine, because most of them are both terrestrial and marine. They're not finding a lot of things that are independent only to this area.
Ah, interesting.
But sounds like there's some interest in, again, not strictly marine, but fungus that are found in the marine environment often have interesting microbial defenses, and so there's interest in understanding them better because they seem to be figuring some things out regarding, defense mechanisms and interactions with the rest of the biosphere. Super cool.
I didn't catch that last move. Are you bringing the ship farther away from the wall?
Yes.
Okay, thanks.
The Argus shots are amazing.
I like it when I just slightly have Herc underneath the rock,
Yeah.
just tucked away. Not in a dangerous way, but in like a, look, I'm hiding kind of way.
Not in a dangerous way, just in a fun way.
Yeah, and an ostrich burying his head in the sand kind of way.
It really looks great when you're backlighting a few of the corals. Those have been some of my favorite shots so far.
Yeah, like light from underneath that you can see from Argus view.
Mm-hmm. Precisely.
Can I get the camera cam, please? Mesocam cam. That's 6-ish. 6 with modifications.
All right, everybody, what are some of
Thank you.
your favorite findings of the day? Question coming in from somebody who's just joining the dive.
Purple cucumber.
Octopus.
Ooh.
Transparent vampire cucumber.
Yes, all great choices.
I found myself.
Bridge, nav, another step.
Were you
Step
lost?
outside, please.
Yeah, I was pretty
Take
lost.
5 minutes. Yeah.
Ooh, there I am. Oh, good.
And a curious Dumbo squid. Yeah.
Yeah, Dumbo
Dumbo octopus,
octopus.
yeah.
Very
Wish it-
cool.
Sure.
Give this guy his 5 minutes of fame.
I'm gonna cheer for the rock that tumbled all the way down the slope right when we first took over the watch.
Oh, yeah.
I thought that was a great illustration of how steep
know.
these cliffs are.
Dumbo octopus again in Argus cam.
It's coming out.
What?
wide.
Behind us.
Is it?
Argus cam's got
Oh,
Dumbo.
yeah.
Oh,
Oh
he's
my
sneaking up
gosh.
on us again.
Oh,
New Dumbo octopus.
New friend.
Or the same
Or the
one.
same one.
Or there's someone
Yeah.
lurking.
Don't go up more. Don't go up more.
Lurking.
I don't want you closer.
Argus, you can. Can you tilt down with Argus?
All the way down.
Okay. Oh, that's okay.
Wow.
Oh, nose there.
Sneaking
He-
up.
He's gonna do what he's gonna do.
Oh, closed his eyes.
He did.
Maybe he'll
Wow.
come home with us.
I think
I hope not.
he's
Yeah.
coming. He's coming to check us out.
That's so interesting.
Whoa.
Checked.
Yeah.
Gosh, I'd love to have a pet Dumbo octopus.
Aren't they so cool?
It would probably escape.
It probably would. Very clever.
Okay, where'd he go? Is he gonna be attached to the thing when we recover?
He's on the greener pastures, I think.
Let's hope so.
Moved on.
Realize we're not as cuddly as we look.
I worked in an octopus lab for a little bit,
Oh,
and
that'd be cool.
one of the problems was they kept escaping because they could learn how to do the locks, and
Yeah.
they'd
So
undo the little
fascinating.
locks, come out, and go switch tanks and socialize. And the thing that stopped them, we found, was AstroTurf. They didn't like AstroTurf.
Really?
Huh.
They wouldn't cross AstroTurf.
Would not. Yeah, they hated
How
it.
do you find that? How do you
Just
discover
keep trying
that?
different things.
Well, when you're trying to do studies on octopus and they keep on changing tanks, you got to try something.
Just go to the hardware store and buy every type of floor covering you can find. Shagrug, no, they love it. Okay,
They crawl out
vinyl.
of the tank, on
Nope,
the ground,
fine.
and then crawl up.
Oh yeah, they'd crawl out. They'd
Jeez.
go to another tank and eat fish out of the tanks, and I mean,
That's so wild.
The Oregon Coast Aquarium had that problem while I was there studying at Oregon State University's marine lab. There's a little aquarium there, and they had a giant Pacific octopus that would crawl out at night and go crawl into another exhibit and eat things and then come back by the morning. And they got the footage on it and proceeded to, sort of triage solutions just like that to figure out.
See that boulder on the edge? That is just itching to be pushed off. It's a thing that's like irresponsible to do when you're hiking, but you really want to.
Oh, that is good sport when you're hiking.
Well, I mean, as long as you're really sure that no one's below you. There's a name for that. I can't remember what it is. Like
Trundling.
Trundling? Yeah. Okay.
I think I told you that though, so we've come full circle.
You've just
Maybe,
created a
maybe.
term together. You're the only ones who know the word.
that's the name for pushing a rock off a cliff and hitting somebody down below.
Not
No.
and specifically not hitting someone.
I think that's manslaughter.
Yeah.
I think like trundle is the anthropogenic movement of rocks downslope.
They're progenic.
Can we
This
have
one looks like
quick
it's been
zoom on
eaten
that
a little
cloud?
bit. Yeah.
Actually,
Ooh, something's on that.
Something's
Yeah.
going for it.
Looks like a crinoid is on
Yeah. Okay. Go ahead and
the
zoom in.
coral.
zooming in.
Hmm.
Zooming in on the limbo bar.
Oops. Ooh. Yikes.
And dropping the light.
It does look like a crinoid.
But did he get stuck?
Can crinoids be parasites?
Or it's a Brazilian sea star. I bet. I
Yeah.
bet it's eating it.
Yeah.
Sea stars are one of the big predators. Miles, you want to see a predation event? Here it is.
Oh, that's Yeah, I
nom,
technically.
nom,
What's the
nom.
coral though?
I don't know.
Oh.
Those are huge polyps.
Yeah, aren't they? They look different.
Seems really different.
You think
Yeah, he's
that's a
cleaning
sea
it
star?
off.
It's
I mean,
stripped bare up to that
Yeah.
point.
Oh, yeah.
I
It
think he's
looks
working
like-
his way up the stalk.
It's a horror movie
I
for
thought
that.
they were filter
that
feeders.
sea stars?
Well,
Brazingets?
I don't know. I'm operating off of. Gosh,
we
I'm looking for
found-
something with polyps that big
we
here
found
in
the
the guide.
carnivorous version.
Bridge nav, another step.
I don't think I have a good answer for you, Sina.
Yeah, no, I'm just gonna write unusually big
Large
polyps. I don't know.
polyps, yeah. Flag this for later.
Trevor, what's up top? Are we gonna?
Looks
We're
like an
going too
anemone.
fast.
We can go up back and check out the anemone.
Okay. It's got an interesting colored base. Drop to light.
A little bouncy, but
Alright.
I kind of came back in a hurry.
Yeah, it's okay. Thank you.
Yeah.
He's a little larger than the others
Mm-hmm.
that we've seen.
Mm-hmm.
I got a question that I'm only 90% sure I know the answer, so I'll ask it. Is algae plants?
Yes.
Yeah.
Okay. Thanks.
You're welcome. We're here for you. Hello to Polly in Costa Calma, Canary Islands. Hope you're enjoying your time off the ship.
Can we zoom on that stalk too when we're there?
This white one?
Yep.
Sure, I'll make that happen. Try and make it smoother than that anemone.
You ready, Trev?
Almost. Okay, go ahead.
Okay, pushing in.
Trevor, I also just realized we totally lied to you. Algae aren't plants. They're
Why
yeah,
not? They are.
they're photosynthetic, but they lack roots and stems and leaves, and so they're not officially plants. They're either
like terrestrial
prokaryotes,
plants.
like blue-green algae, yeah, or eukaryotes. So they're their own thing.
Wow.
All right.
So no
Wide.
cellulose. Cellulose is now the defining factor of a plant.
If they are cellulose, there's no cellulose.
No cellulose. That was kind of a question more than a statement. It's definitely a question.
I mean, I think it's kind of, algae covers so much, down from like the little single cellular ones to the big ones.
Bridge
So yeah,
now.
some algae are, I guess, better characterized as like
20 meters, bearing 040.
protists or bacteria.
Let's go down and check this guy out.
There's a mushroom coral up there too.
Yeah.
So I thought photosynthesis defined plants. That's not true?
I don't think so. So I mean, things we call algae are all over the place, from like cyanobacteria or like diatoms that photosynthesize, are chromista, which are more like kind of their own kingdom, like more protists.
Coral.
Yeah.
Yeah, zooxanthellae.
Coral?
So like I mean, I'm just reading because there's a lot written about this. That's like, well, yes, no, but really no, but it depends. So, aquatic multicellular algae definitely look like plants. They have leaflets and they have, the same kind of, cellular tissue. And it's likely that land plants came from them, is what we've got here. But recent phylogenetic analysis placed red alga as a sister group to all other plants, starting with green alga. However, some phylogeneticists take a broader look and lump red alga in with plants, but not green alga.
Well, seaweed or macroalga.
What's this little fluffy looking thing? Maybe that's just sediment. Never mind.
Is that little TNF4 floating by?
There's a really long one.
Yeah.
Oh yeah. Yikes, that's...
It's got a big base, too.
It's got a huge base.
That's what it's all about.
Oh, no.
Oh, no.
I'm gonna go put lasers on that base when I can get a little closer to it.
No trouble.
Or is that a sponge right
That's
beside
horrible.
it? I think it's a sponge right beside it, guys.
Yeah, I think you're right.
Thought you didn't like puns, Pauline.
If you can't beat them.
That's the spirit.
Join us.
That is not a base at all.
Yeah.
No.
That's not a base at all.
Optical
Oh, it's one of those really
illusion.
cool sponges where they have a lot of those Holta spicules out on the outside. They look all hairy.
Something else entirely. Which we will zoom in on
So I'll let
Bridge
Shinsky
nav,
and
when
I.
Trevor's ready.
That's cool.
You ready, Trev?
Sure, you can go in halfway and then keep it in
Okay.
frame.
Let's see, we're gonna need less light.
I am so disappointed. I brought the wrong notebook, and last year I had the chance to sell with Dr. Chris Kelly, who is extremely passionate about sponges and gave, a wonderful identification class in the lounge throughout our NA101 expedition up into Papahanaumokuakea National Monument about how to identify all of these sponges, and I forgot my notes. So
Okay, come on.
just
Come on,
bummed.
wife. We got a good shot of that. That was
Yeah.
really nice.
Hopefully somebody can help us with the ID there.
We seem to have a lot more stuff in this little area.
Yeah. Well, we're coming down to this little promontory. I debated whether to cut across the top of it or come around.
Do we want
this
to
thing.
fire off a Niskin here?
Well, I think, yeah, why not? We have plenty.
I think we're only going to take, was it 4 or 5 per dive?
6.
We have 6 on board, but I think there was a limited amount of filters. That's how I understood
I
it.
think it was 4.
'Kay.
Or not more than 4.
Okay, for the whole cruise?
No, for the whole each dive.
Okay.
And
4
we
discus per
yeah,
dive, yeah.
and we can if we don't take them on one dive, we can carry those over, sort of thing.
Push in, Trev.
Sure.
But we've just
no
taken
hands.
one. Is that
Bridge.
right?
Yes,
nav,
that's it.
Another step.
Heaps of space.
Heaps.
It looks like someone stuck it on the wall with gum.
Yeah.
Chewing gum or something, yeah.
Or like that poster putty.
Oh,
that
yeah,
just
yeah.
is never really that sticky, no matter what.
Dive plan says 6 Niskin water samples may be taken at each dive site.
Yeah, it was a change after we realized we didn't have all the DNA filters.
Roger.
Yeah.
I just know what I read.
Yeah. It was a call Meredith and decide how many we want to do this whole expedition and then backtrack to how many we want to do.
eDNA filters are a special kind of filter.
Coming wide.
Yes.
We tried using coffee filters, but it didn't quite work.
That's why my coffee tasted funny.
Was that a bamboo coral?
Yeah, I think it was.
Can you brighten it up a little, Wayne?
Yep.
So you know what's the size on the eDNA filters?
Thanks.
They're little,
call 'em
little,
little,
right?
Wayne.
The size?
Mm-hmm.
Brighten it up a
The
little.
pore size?
The
you
pore
mean?
size, yeah.
Oh, I don't really know that.
Okay.
All right, I'm going to come up the wall a little bit.
If they were standard filters, we might be able to trade Palmyra for some gasoline for filters.
See if I can pull it up
Oh, that's right. Are we doing that tomorrow?
tomorrow.
Yep.
Mhm.
Cool looking sediment, or not sediment, rock here.
Yeah.
Mhm.
All kind of folded. It almost looks like someone threw an old carpet down.
Mhm. Yeah, really cool.
Okay.
Not big an enemy.
It's cool. Pretty sonar target. This looks nice.
Sea prints too.
Yeah.
I'll put that out on.
Big shrimp. Or is that a fish? What
Yeah,
is
that'd
that?
be great.
There's a fish. Let's zoom
Push
in,
in.
please, and keep him in frame.
Okay.
What is that?
That's a fuzzy thing.
Yeah. A sea caterpillar. It's not a
Is
land
that a
caterpillar.
siphonophore?
There's no propulsion module.
Slurp gun.
Propulsion module.
Yeah, slurp it?
Really?
I don't know.
We could try.
Let's slurp it.
Come wide, please.
Nothing else you
Coming
can
wide.
slurp.
I wouldn't get your hopes up, but
No.
we can
Uh-oh.
try. Okay, could you give me bucket camera on her cameras? You'll have to turn bucket on the
Oh.
screen.
Challenge.
You have to turn
accepted.
starboard rail off. And then let's cycle it around to whatever jar we want. We should adjust that camera too. So hydraulics page,
Come back, Shane.
sample jar forward and reverse. Looks like we're on jar 6 right now. I suppose that doesn't really matter. We could go in any one, can't we? That's jar 5. Okay, let's.
Tuna nose.
That's 4. That's 3. That's good there. That's fine. And then let's do starboard suction at 40%. That's this one here.
Okay.
And you're going to need to watch your delta.
Is it 4? Slurp jar?
It's slurp jar
3.
3.